In this lesson, I want to look at the physiology that's happening in the neuromuscular junction. Remember, this has to do with muscle physiology, and in muscle phys, we can break it down into 4 big steps. The first step that we discussed was the action potential generates in a neuron. Well, here we're seeing step 2 in great detail, that is the neuromuscular junction, or the synapse. Step 3, the action potential generates in the muscle cell, and step 4, the muscle contracts. And we're going to break those things down and look at these in great detail, but just note ourselves here that we have a neuromuscular junction set up here. As we look at this, we have a neuron, a muscle cell. Well, the neuron is the presynaptic cell in the synapse. The muscle cell is the postsynaptic cell of this synapse, with the space between the 2 cells called the synaptic cleft. Well, in this, we want to give ourselves, in the neuron, we have to get this cell ready to do what the cell needs to be done. So I need vesicles, drawn in blue, with acetylcholine, drawn in red. ACH, acetylcholine is the transmitter that the neurotransmitter that we need in order to make the muscle cell work. That's why we think of the neuron as the boss, the muscle cell as the employee. If I go to the muscle cell is an excitable cell. And it does possess a very special protein bound throughout. It's an integral protein bound to the cell membrane called the sodium-potassium ATPase pump. I've drawn this in blue. The sodium-potassium ATPase pump has a very simple job. It will bring in 2 potassium cations and kick out 3 sodium cations, and this sets the muscle cell to that of resting, and the resting is at negative 95 millivolts, and that is done by the sodium-potassium ATPase pump. But let's further look at this concept of the sodium-potassium pump, because this is a very important thing. If you know, because it brings in 2 potassiums and kicks out 3 sodiums, that sodium is in a high concentration outside the muscle cell, but low in. Potassium, on the other hand, is in high concentration inside the cell and low concentration outside the cell. This is critically important to understanding the physiology of this cell, and we'll look at that later. So we got to remind ourselves of this. So as we go from resting, we're going to start kicking this guy in and begin looking from resting state. We're going to go to other states. So I'm going to erase some things here so we can clear ourselves out. We'll turn the sodium-potassium pump off. And this will give us a good starting point for what's going on. Now, remind ourselves that the neuron only works when there's an action potential will propagate. So an action potential will generate on the neuron. I'm going to draw this in a little bit of an orange. We don't have yellow on this whiteboard app. But as this comes across, as the action potential generates down this neuron and gets to the terminal end, at the synaptic end on the terminal buds of the neuron, the action potential sweeps down. Now this opens voltage-gated calcium channels, and calcium will rush in Ca2 plus rushes in. Now this results in exocytosis. Basically what happens is the vesicles containing acetylcholine will move forward, and they will release acetylcholine across the synaptic cleft. Now, it makes no sense to have a neurotransmitter if they don't have something to bind to. So on the postsynaptic cell of the muscle, I'm going to draw these little Y-shaped little receptors. These are nicotinic acetylcholine receptors. And acetylcholine will bind to the nicotinic acetylcholine receptors, and this is where things begin to get interesting. During this state, when the acetylcholine receptors, or nicotinic acetylcholine receptors, are bound to by acetylcholine, you will open up the sodium gates. Now, just for special purposes, I'm drawing them separately, but remember, they are actually the same thing. The receptor sites are on the sodium gates. These are chemically gated ion channels, and sodium will rush in, Na+. Now, please note, it does this for two reasons. One reason is because sodium is in high concentration outside the cell, and sodium is in a low concentration inside the cell. Now, if we look back, our sodium-potassium pump set this concentration gradient. But one thing I didn't tell you is, beforehand, the cell was slightly negative inside. Because there was less sodium in, there was actually a very low concentration of potassium. Essentially, there is less potassiums in than there are sodiums out, which left the cell being negative on the inside and being positive on the outside. So we were kind of saying that before this point, the cell was being called polarized. As potassium, as the sodium-potassium pump shuts off, it sets into a polarized state. Sodium rushes in, not only because it's high to low, high out, low in. As it rushes in, it's attracted to the slightly negative inside of the muscle cell. As this happens, the cell gets to be slightly positive. As the sodium rushes in, it becomes highly positive and keeps getting more to that as it will reach threshold. And then it will go from threshold to the action potential. And this is how step 3 happens. Action potential is generated by the massive diffusion of sodium into the cell because I open because the nicotinic acetylcholine receptors are bound to. Sodium gates open, sodium rushes into the cell goes from negative to positive, it becomes depolarized. A depolarized cell is experiencing the action potential. And this is where things get the most complex for us. So by this point, the extracellular calcium has rushed in because of the action potential in the presynaptic cell. Acetylcholine is released. It binds the nicotinic acetylcholine receptors. That opens the sodium gates, and then things get further interesting. As the sodium is rushing in and the cell gets more positive, an action potential can form. But I need to show you guys one more thing. I'm going to draw in a triad. Remember, a triad is made up of one T-tubule here and two terminal cisternae that I've put with the dots. So there are one T-tubule and two terminal cisternae, a triad. Now, the terminal cisternae store calcium 2 cations. Remember, if calcium is inside the terminal cisternae, to say in a high concentration in the terminal cisternae of the sarcoplasmic reticulum, the muscle is relaxed. Right now, this is a relaxed muscle. Now, when an action potential generates down the muscle cell because it gets depolarized, it will go down the T-tubules. This opens the voltage-gated calcium channels in the sarcoplasmic reticulum in the terminal cisternae, and calcium, Ca2+, leaves the terminal cisternae. That results in muscle contraction. You guys have learned this later on, how this actually does result in muscle contraction, because this will result in the change in conformation of the troponin-tropomyosin complex. As that occurs, the muscle is now contracted, depolarization is done, and we no longer need the sodium gates. When we get to a point, sodium gates need to close because we've contracted and we need to stop. There's a way we do this. Now, we can use various colors to do this. Typically, we use a yellow, but you see my colors are limited in this palette. So I'm going to use a gray here, and I'm going to color in a little enzyme here known as acetylcholine esterase. Acetylcholine esterase cleans off the nicotinic acetylcholine receptors. It removes acetylcholine. It breaks it down into choline and acetic acid, essentially. As that happens, we can recycle this to make more acetylcholine. As we break that down, the voltage-gated, or the chemically gated sodium channels now will close. Now, we'll get rid of the triad. We no longer need that stuff. This cell is now done depolarizing, and we go from depolarization, and we're going to show you how we repolarize the cell, how we get it back to negative. Now, the first big step, after acetylcholine has been cleaned off by acetylcholine esterase, and the neuron is back to resting, and it goes through, you guys will learn how that happens later on. We get our acetylcholine gets back into these vesicles. They stay back in. This guy is ready and go back to resting state. And you guys will learn more about how that happens. And I'm going to erase all this just to clean that up. And we'll talk about that more in Chapter 11. Now, with nicotinic acetylcholine receptors there, the only thing we got to do now is we will have all this potassium in here. We want to let it out. So we have voltage-gated channels to let the potassium out. Potassium leaves the cell. Remember, it does this for one reason. There is a high concentration in here and a low out. It leaves for that reason, and that's the only reason we really think about it leaving here. And as that leaves, the cell starts getting more and more negative. It got highly positive. When we leave all these positive ions out, we then start getting this cell to be more negative. So I'm going to draw this in as being a negative cell, and we start repolarizing. It becomes repolarized. Now, to further reset this, our good friend, the sodium-potassium pump, needs to kick back into gear here. And the sodium-potassium pump will reset the transmembrane potential. It brings in 2 K pluses, kicks out 3 Na pluses, and that resets it back to resting, and the cell's ready to go again for another contraction. So all the big steps: action potential generates in a neuron, the synapse, the action potential generates in a muscle contraction can all generally be discussed here in this little diagram. And hopefully you find this is a helpful little lesson to understand this very complex physiology. And this concludes my lesson on neuromuscular junctions. And I hope you guys found this little lesson helpful.